Hola. Now? Yeah. Welcome again, you. Everybody, we are here at the Pasteur Institute, which belongs to the International Pasteur Network. And so we are a research institution, non-profit organization, which are guided by the One Health approach. So that's mean environmental health, human health, and animal health. And the institute is organized mainly in core technology or shared research labs and research labs. And so one of the features that has our institution, in general Pasteur, that are really commitment to training and hand-on training course. So then there's an important part of education within the institution. So that this course is part of, this workshop is part of this training initiative. So the workshop in numbers. So we have the registration open for 1 month. And so we had more than 200 registration. So you are lucky that you are here. So 25 participants were selected among these more than 200 applications. And the selected participants are from 12 different countries, Latin American countries. And we have 7 invited speakers, really good speakers, so get profit from all those speakers that are spending the whole week here. Plus 3 local organizer and speaker and secretary, and Paula, Karen, and myself, and of course Natalia, who was helping a lot with the logistics. So you know Natalia per email and now during the registration. So we are here thanks to the Natalia's help as well. And so the workshop will be organized in theoretical lectures. This theoretical lecture will take place here in this room, Salón de Actos Guillermo de Guiero. Then we will have a tutorial or data analysis part that will take place in Sala de Informatic, or the Informatic room, that is on the back part. So everything is in this floor. We don't have to run, and so everything is on this floor, unless that the cantina is 1 floor downstairs. And we will have wet labs, so, and will take place simultaneously at the Laboratorio de Enseñanza, that is also back here, Laboratorio de Equipos Comunes, Lab 39, and Laboratorio de Clasificación Celular, Lab 30. So where are the different cytometers are installed? So here we have the Cytec Aurora, the spectral cytometer. Here we had the iTune, a conventional cytometer, and in this lab we had the FACS area fusion. But you will guide to this lab, and also if you get lost, there's a stamp or printing in each door with the logo of the course. So in case that you get lost, and we are going to use lab code, blue lab codes. So in a way that if you get lost, you can ask the other people where to find, so they identify you as a foreign lost in the middle of the course. We have some lockers. I think only one asks for a key, but during the course, if you want to put on a safe place all your stuff that you are not going to use during the course, we can use the locker that is behind. You will receive a key, and at the end of the course, please return the key because the course move over and over. And if you didn't realize, or then later on you think, maybe it's useful to have the key, so please go to the registration desk and then ask for a key, and they will write your name, and you get the key, and then the locker is here. The most important thing, Wi-Fi network is Institut Pasteur, and the password is Montevideo, but it's not in capital, it's in low. I think I tried to put in capital, so it's Montevideo, everything in cursiva. Lunch will take place at Institut Cafeteria, that is 1 floor downstairs. We will go together. And the workshop support material are already on the classroom. Probably you had received an invitation to join the Google Classroom. So if you don't receive, please let us know, and then we will send you the invite. And one important thing, a social activity, just keep away. We will have a dinner together on Thursday, and we will start from here to the place where the dinner takes place, that is Aras Cuatro Piedras. And then the bus will return to the hotel. And so those from Uruguay, then you had to move to from the hotel to your places. So here is the program. It looks quite tight, but in general, during the morning, we will have the lectures and then the break, and then in the afternoon, the practical or tutorial session. I forgot to say that the language of the course will be, let's say, mainly two, English and Spanish. Don't be shy and ask every time that you want to ask for translation, for explanation. I will do the introduction in English, but then I will switch to Spanish, but because I would like to welcome Zosia and Bill in English, but later on, perhaps we can switch to different language, but don't be shy and ask all what you want to know. So here is the program you have also there. And so we are here thanks to the founding, of course, because otherwise we cannot be here. So first, the first grant that we got is from our national agency, just to bring 3 speakers. Then we have a grant from Unu-Biolac, which support the fellowship for all Latin American, the 15 Latin American students that bring you here and pay your ticket and your accommodation and your lunch. Then the ISAC, International Society of Advanced Cytometry. We had the pleasure to have Zosia and Bill. Zosia is the chair of the Life Education Delivery Task Force, ISAC Life Education Delivery Task Force, and Bill is the co-chair of this task force. And also Rosana is working on the education committee of ISAC as well. You are also an ISAC member. And Lola also is working in a SRL content. So then we had the pleasure to have all these people here. And thanks to ISAC support, we have Zosia and Bill here. Then Pasteur Institute with the logistics and some money, and also Mercosur FOCEM resources that provide Mercosur, Argentina, Paraguay, Uruguay, and Paraguay support to do this type of training activity. And also we had some sponsors, so Genomedica and Cytec. Genomedica is our local distributor of Cytec. And also we have support from BD, from the, you had received the invitation for the flow shop license. If you don't get this, if you didn't get this information, please let us know, contact Paula or Karen. Later on we can figure out how to receive, or we can use other Skype to use the flow show during the tutorial. And also we got some slingshot particles as a demo to use on the flow cytometers. And also, in addition, just to be aware, in the classroom, there's a, I think, 15 minutes video from BD of using flow show to analyze spectral data. So this video, it's on the classroom, so you can watch whatever you want, or perhaps you wait until you receive all the lecture in spectral cytometer and then get involved in flow cytometry and flow show, and then put, but it's a video, so it's on the classroom material. And saying that, I think, yes, very important. One of our financial, two of our financing agency asked if you want to post or do some advertise of the cost, what are interested and so on. So probably if you use Facebook, LinkedIn, or Twitter or Instagram, don't forget to put hashtag FOCEM and Unu-Biolac. Or if you write something, just ANII, ISAC, Unu-Biolac, Institut Pasteur, and so we are all happy, or the founding agency are happy as well. So try to spread the work, and I think with this, I think I have something with mentivity, but perhaps I can avoid this, and later on we can do it in a way that we don't lose so much time. I'm saying that perhaps I can, there's two possibilities. We would like to the student to introduce themselves. So the idea is to say your name, institution, if you are using or you are independent user of the flow cytometer or you don't have knowledge, and what are you looking for this course? So for which purpose or what are your intentions? So perhaps we can, I don't know what's the time now, if we have some minutes or for the presentation, or perhaps we can start with Zosia and at the end we can start with half of the student and then after the coffee break, half of the other students. So that I will write the question that you had to say name, institution and country, if you are independent or not independent user, and for what are you planning to use or the benefit of this course for you in your PhD or in your core facility and so on. So don't get nervous or stressed right now. So just at the end of the talk of Zosia. With this, let me switch. I will switch for Zosia introduction and then yes. So we will move to our first talk today. The first talk is going to be delivered by Zosia Maciorowski. I never say yeah. Zosia received her bachelor in microbiology from McGill University in Montreal and a master in biology from Wayne State University in Detroit. She has worked in many labs and countries over the years on a variety of different subjects, from the early stages of tissue culture to small animal surgery, monoclonal antibody production, and early immunological and molecular biology techniques. In the 80s, she specialized in solid tumor preparation for multicolor and cell cycle analysis. For the last 20 years, she has been responsible for the flow cytometry core facility at the Curie Institute in Paris, France. And Zosia has been involved with the International Society for Advanced Cytometry for several decades, contributing to the Congress program, leading pre-Congress workshop, and at the present, as I mentioned before, she is the chair of the Live Education Task Force of ISAC with the mission of delivering and managing innovative live, in-person, or interactive education workshop across the world. Zosia has led countless workshops. I don't know if you had counted, but yeah, several workshops, lecturing and giving practical sessions in workshop and hand-on training course in Europe, Asia, America, and Africa. And this is the first time of Zosia in Uruguay, Montevideo. So we are welcoming you, Zosia. Please, the stage is yours.
So the ISAC talk first and then...
Yes.
Okay.
So, si tienen alguna consulta, pregunta, levanten la mano y la pueden sacar para la vuelta en portugués.
Okay, I think this is good, right? Okay. When I was struggling with this, I hit the button, turned it off. Not a good start. So it's a thrill for me to be here in Montevideo. I want to thank Mariela and Paula and Karen and Natalia, who have done an amazing job organizing this workshop. One of the best organized workshops I've ever seen, and we've done a lot of them. And Mariela took us around Montevideo yesterday and showed us its wonderful city. It's a beautiful place to live. So it's a real pleasure being here. I'm going to talk a little bit before we start the cytometry bit. I'll talk a little bit about ISAC because I've been an ISAC member for a very long time, and so have Bill and Roxana, and as Mariela said, everyone else here. So ISAC's been around since 1978, when the first flow cytometers started being popular. We have about 1,600 members worldwide, so it's not too big, it's not too small. The members are flow and image cytometrists. And we publish 2 journals, the Cytometry A and Current Protocols in Cytometry is also part of ISAC. And the membership are academic, about a third academic researchers, a third SRL. SRL is the new word we use for core facilities. And then we have cytometrists working in pharmaceuticals and industry as well. So we have an better. Okay. And Mariela, I'm changing the slides here. This is right. Is that right? Change the slides here? Or is there a gizmo?
It's fine for changing, and this is the last.
Okay. So we have about 1,400, 1,500 attendees at our annual Congress. Last year it was in Montreal. This year it's going to be in 2024 in May in Edinburgh in Scotland. And so it alternates. It goes from the West Coast to the East Coast and then to Europe. And we've also had one in Asia. We had one in Singapore, and we're hoping to have another one in Asia maybe in 2025. So it's where all the cutting edge cytometry is. It's for academics. All of the core facility SRL people are there to see the new instruments, everything on display. The vendors have all the latest instruments there. And there's a lot of tutorials and workshops on the basics and on the latest topics. And networking. It's a nice bunch of people at ISAC. They're all very friendly. The oldies from the 70s and 80s. We're starting to lose them, but they've all been very approachable. You could walk up to somebody like Howard Shapiro and he would tell you all about his work and sing you a song about how he lost his laser. So it's a really, really nice group, and it's a good place to find the people who are really the experts in your field and also to start networking with them. It's a very kind group of people. There's an Emerging Leaders program, which is interesting for young people. They're competitive programs. They're quite competitive for young scientists to develop their career. And we're developing future leaders in ISAC as well. So we're grooming people for the next generation. There's a 5-year membership in ISAC with the journal. You're funded for travel to Congress and funding for mentorship programs. And then there's opportunities to participate in the different committees. And there's 3 different versions of this. There's the Scholars Program, which is geared toward academics. There's the SRL, or Shared Resource Lab, or Core Facility Emerging Leaders, for those are the people that are running core facilities. And then there's now a new one called the Innovators Program, which is geared towards young scientists who are expanding knowledge and developing new technology and innovation. And so you can take a look at the ISAC website and find all of this. There's online education if you're a member. There's webinars, there's courses, there's recordings at Cyto University. And we have the Live Education International Workshops, which is what this is. It was originally founded by Dr. Attar Krishan. He's also a co-chair. He started these in 2002 in India. He's from Kashmir, and this was his baby. And he then developed it over the next 10 years, expanded it into Turkey, China, the ASEAN countries, and we've been doing them on all the continents now. And it's taught, the workshops, as you can see, have theory and practical sessions. A lot of them are hybrid now, so they're being broadcasted as well for people who can't make the actual workshop. Taught by the international and local faculty. And we have a range of topics from basics to advanced clinical and research topics. And it's really the host at the institution that chooses what range of topics, what level, and what topics they're interested in and are most useful to their students. And so the vendor sponsorship and the local grants are really important. We couldn't do this if we didn't have the vendor sponsorship. So we're always really grateful for them, and also for all of your local grants. You've been, your government has been very generous with your grants. So we're really lucky about that, and lucky to be able to bring you here from all over Latin America. These are the workshops from 2002 to 2023. So all those little dots are where we've done workshops. And this is the second one in South America. We haven't done as many in South America as we should have. And so in the next few years, we're hoping we'll be doing more of these. And this is the actual list of all the workshops. So during COVID, you can see we went hybrid because we couldn't, we went virtual over here. So all of these ones were virtual, and these are all up on a website that Bill has at NIH. And so you can access the recordings of all of these different workshops freely, and Bill will give you the information for that. But there is a huge amount of material up there on all kinds of topics, both clinical, basics, advanced. We've got high dimensional data analysis that was done with Jonathan Irish and his crew. And Maria did a week-long spectral one a couple of years ago with the South African workshop. So there's also an imaging flow cytometry one. So there's really, it's worth taking a look through all those recordings on Bill's website. And so now we're doing live, virtual, and hybrid where we can. And we're also in conjunction with the ISAC now has a committee called the Instrument for Science, and they organize donations of instruments in different places. This is Bill's baby. There's been 30 instruments donated around over the last 20 years around the globe. He's installed most of them and serviced most of them. And the education, live education tries to do the training for this as well when they're installed. There was just one that was installed in Paraguay, and Jose is working with one that Bill just installed in Ecuador in September. So, and the person from Paraguay, I believe, is here as well. Yes, there. So he was in Paraguay installing in August. And so I think there is another one going in. I can't remember where, but he'll be able to tell you more about it. And so these are our emails if you want any information that we can tell you. The ISAC fees for joining ISAC, there are special fees reduced for a lot of countries that are resource limited. So it's less expensive than people in North America would pay. So depending on which country you're from, there'll be a different rate. So let us know if there's anything that we can help you with. Thank you. Yeah. Great, thank you. Okay, I'm going to start with the basics. So if I'm talking too fast, I'll try and go slowly. This is very, very, very basic. So those of you who are more advanced, you might be a little bit bored. You can go back to sleep. And I'm going to talk about the basics of fluorescence and how the instruments work. So what is flow cytometry? It's a technology which allows us to measure light scatter, fluorescence intensity on cells or other particles, and one by one. The cells must be in suspension. We originally had thought that we might be able to put subtitles up, but we're not allowed to, we can't do it with this particular software. We were hoping to have subtitles in Spanish here, so I'm sorry. Sorry, my Spanish is non-existent, but so the advantages of flow cytometry are the speed. You can measure millions of cells so that you've got good statistical significance. It's multi-parameter, and you can sort the cells at the end to get pure subpopulations of the cells that you were interested in or the researcher is interested in working with for further experimentation. So when do you use a flow cytometer? You want to know when your cells are big, your cells are small. You want to know how many of your small cells might be red or green. or red and green. And so cytometry, flow cytometry has a lot of different applications that you will use this for, research applications and clinical applications. And I think that there's an applications talk that Bill's giving, so I won't go into detail on this, but you can see here that there's quite a large range of different cellular biology, but also plant biology, marine biology, microbiology, extracellular vesicles are now a very big thing. And of course, in the clinical, there's a huge range as well, and particularly for leukemia and lymphoma typing. So I'm going to talk about light, what we measure, fluorescence and light scatter, and how a conventional flow cytometer works. You will get more information from Maria on the spectral flow cytometer, which is the next step up from conventional, that gives you some advantages that conventional doesn't. So we work with light in flow cytometry, and we work particularly with wavelengths that are between about 300 now, and we're going above 800 as well. So we're in the visible and down into the ultraviolet range here. And if you remember your basic high school physics, the shorter wavelengths, so this around 400, 350, are very high energy. And the longer wavelengths are lower energy. And so remember this high energy to low energy transition that will give us fluorescence. So a fluorochromes are molecules which can absorb light at one wavelength, and then they re-emit that light energy at a longer wavelength. So they absorb it at one energy, which is probably higher energy, and then they re-emit it at a longer wavelength, which is lower energy. And the structures are generally aromatic rings. So this is fluorescein or FITC, or FITS as we call it, which you're probably used to seeing both in microscopy and flow cytometry, and phycoerythrin, series of rings. And so how does this work? This is a very basic schema of what a fluorochrome does. So you have an electron that's a resting level, and it can absorb the light at a certain wavelength. It absorbs the energy, and then it will go up to a higher energy level, which is unstable. And so that higher energy level, it will lose a little bit of that energy non-radiatively, and the rest of that energy, it re-emits as fluorescence at a longer wavelength with lower energy. And so this would be a schema for FITC, because it can absorb wavelength at 488, and it re-emits around 532. And so this is a really simplified little schema. In general, it's really the fluorochrome's electron cloud that absorbs and re-emits the energy. For each fluorochrome, it has its own excitation spectrum. So each fluorochrome is capable of absorbing light energy over a specific range of wavelengths. So here, FITS can absorb energy all across these wavelengths. It's got an excitation maximum where it absorbs most efficiently, but it absorbs across its whole spectrum to more or less efficiently. Similarly, it has an emission spectrum that's specific for the fluorochrome, and it has an emission maximum where it will emit most efficiently, but it will emit across a whole range of wavelengths. And so you can see that 520 here is its emission maximum, but it will emit below that, and it will tail off into the higher wavelengths. This is very typical. Fluorochromes tail off to the right, and this will affect spillover and spread and compensation, and we'll talk about that in more detail later. So we use lasers on flow cytometers to excite the fluorochromes. The lasers that are found on the standard flow cytometers, first we usually got a 488 laser, which is a blue-green laser. A 633 laser is also very standard. These are the red lasers. The newer instruments now have a violet laser, a 407 or a 405, a yellow-green laser, and we'll see why in a few minutes. Yellow-green lasers are generally around a 561. And on the newer machines as well, you'll get a UV or a near UV laser. And so this allows a whole range of different fluorochromes to be excited and measured. How do we use the fluorochromes? We can use standardly in immunofluorescence that we use an antibody that's to an antigen on the cell, either on the surface or inside the cell, that we will couple to a fluorochrome, and that will allow us to quantitate the amount of antigen that's on the cell we want to measure. So here we've got one antibody that's coupled to a green fluorochrome, one antibody that's coupled to a red fluorochrome, two different antigens, and so you can measure both these different antigens. Another major application that's used are reporter genes, where you're transfecting a cell with a gene that you're interested in knowing what it does, and at the same time you transfect a green fluorescent protein or another fluorescent protein. And so the cells that have taken up the gene of interest, that are expressing the gene of interest, will also be that color. So you can then have maybe very low transfection efficiency. Maybe only 1% of the cells took up that gene. But if you have a cell sorter, you can separate out all of the cells that took up the gene and have a pure population of cells that are expressing the gene you want. And then you can go and do your further experiments using a pure population. And then there are a lot of more of fluorochromes that are specific for different parts of the cell, for DNA, pH sensitive, organelles, calcium flux, and a bunch of different others. And Bill will talk more about those. The other thing that we measure besides fluorescence is light scatter. And light scatter is the diffraction. It's a physical property of the cell that refracts, or it scatters light in different directions when it passes through the laser beam. And so the light that it scatters is going to be the same wavelength as the laser beam. And it's scattered in all directions, but we measure it at two different angles. So we measure it in the forward scatter, and we measure it at the side scatter. The forward scatter, so if your laser beam's coming from there, I'm a cell, I'm coming up in the fluids one by one like that. I'm scattering light all around. Forward scatter is collected here in the axis of the laser beam. And the side scatter is collected at 90 degrees. So all of the side scatter in the fluorescence is collected by a lens at 90 degrees. And so what does this light scatter tell us? The forward scatter is roughly proportional to the size and the cell surface properties. So a small cell will scatter less light than a medium cell than a large cell. This depends also on the refractive index of whatever it is. So if you have like a 2 micron or a 7 micron cell, the refractive index is going to be different from a 7 micron bead because it's not the same properties. And so you can't actually use a 7 micron bead to say, okay, that is the same size as my 7 micron bead. That cell must be 7 microns. But it's still useful for very other things, for comparing the sizes. And we'll look at that in a minute. The side scatter is more affected by the structural complexity inside the cell and the granularity. Neither of them they can be used to actually quantitate the size of the cells, but they can be used to distinguish, and we use it regularly to distinguish different types of cells. And you probably are used to looking at, this is the forward scatter on the x-axis, and on the y-axis we have the side scatter. And so down here we've got blood. We've got your lymphocytes, your monocytes, your granulocytes. Up here that will have your cells that will have a lot of internal complexity. And so you can separate out those cells depending on the forward scatter and the side scatter and look at those subpopulations separately. So a cytometer has got a lot of stuff in there. It's not a black box. And it's important to understand what's inside your cytometer so you know that what's happening inside is going to affect your data. So we're going to talk about what's happening inside the not black box. This is a fax caliber. And so a number of you have fax calibers, but the fluidics on the newer instruments like the Fortessa, it's exactly the same fluidics, the same flow cell, and the same system. The collection optics may be a little bit different, but this is still very much how it works. So we're looking at the 4-color fax caliber here. So what do you find inside the flow cytometer? So we're going to look at these one by one. We've got fluidics, which position the cells so that they flow one by one through the laser. We've got the optics, which separate the light emission from the different fluorochromes on the cell and direct them towards the different detectors to quantify each color. And then we've got the electronics. The detectors of light then convert the amount of light that it sees, the amount of fluorescence emission, will convert it to a voltage pulse and then a digital number, which will then be saved in a file. So first we're going to look at the fluidics. So this is Bill's slide, and this is a very simple schema of how a positive pressure, and most of the instruments are still using a positive pressure system. So you've got your sheath tank here. You've got a waste tank. This is where your sample goes. Your sheath fluid can be a saline solution. It can be PBS, it can be water. A lot of them are using water now. For a cell sorter, it needs to be something that has salts in it because it has to be able to charge the droplets. But basically, a lot of the newer instruments are using water. So you connect the sheath fluid to the flow cell, the sheath chamber to the flow cell, and then this also gets connected to the waste. We have air pumps that are going to pressurize this sheath tank. So it's going to push air pressure here. It will push the sheath fluid out into the tubing, and then it's going to inject it into the flow cell. So the sheath fluid is coming up like this. This is the flow cell where the cell is going to pass through the laser and the fluorescence will occur and be measured, and then it'll pass into the waste. The sheath pressure is regulated very carefully. It has to be very stable. We also pressurize the sample port to push the cells through the system. And so we have also a sample pressure here. So this tubing is going to pressurize this tube, and it's going to push the cells up through this tubing and inject it into the middle of that sheath fluid so that it's going one by one right through the middle of the laser. And we're going to look at that more carefully. So this is really very simplified. Most of the commercial systems have complex pressure regulation because it's important that the pressure is very steady, and it carefully controls the sheath and the sample delivery. This is what it looks like in real life. So an LSR2, a Fortessa caliber, this is the flow cell. So your sheath buffer is going through here. This is your flow cell here. Your sample is coming up through here. And with the laser on, here's your laser going through here. Your cells are coming up from here. Your sheath is going there. And your cells are going one by one past the laser here. This is your forward scatter. And behind the screen will be your 90-degree scatter collected and your fluorescence. There are a number of different ways. We've just looked at the positive pressure. Most of the instruments use the positive pressure system. The LSR2, the Fortessa, the Caliber, the Galios, and most of the sorters use a positive pressure air system. The syringe pump can also push through either sheath fluid or cells. So the Guava, the Attune that we're going to be using this afternoon uses syringe pumps, and the Novosite as well. And there's also systems with peristaltic pumps. So the Accuray, the Cytoflex, the sheath for the Novosite, the ZE5, these are pushed through with the peristaltic pump. So while a lot of the instruments are positive air pressure, and the sorters are usually positive air pressure, although the new Cytoflex is a peristaltic, I think, and the Aurora has a different system. This is how we push it through. So the cells are going through one by one, and we want them to go through the middle of the laser beam. The laser is here. As we've seen, here's your sheath fluid coming up through here, coming up from the bottom. Here your cells being injected into the middle. The laser is going through your quartz flow cell, which has a lens behind that's going to collect the light. Your forward scatter will be, where'd it go? There it goes. Your forward scatter will be over here. If you have a sorter, it comes from the other direction. So on the FACS area of fusion, it's slightly different from this. This is a pure stream and air sorter, but you have a nozzle with a certain diameter that then forms a stream with droplets, and the laser intersects the cells in air, in the stream and air. So the fluorescence has to travel through air before it gets to the lens, so it's a little bit less sensitive in terms of quantifying the fluorescence. And so the hydrodynamic focusing, pretty much all of these use hydrodynamic focusing. The Attune uses acoustic focusing, which has definitely certain advantages to it, and we'll see that this afternoon. The cells are injected into the center of the sheath fluid so that they'll be positioned in the center of the laser, and we'll see why in a few minutes. And so your cells are coming through one by one. The hydrodynamic focusing is making sure that the cells are being centered, pressured into the center of that stream. So the sheath pressure remains fixed on a flow cytometer. It's controlled by the flow cytometer. But I'm using this here.
I didn't know before.
Okay.
And some water.
Okay, thanks. Great. Okay. No, I don't need a glass. The bottle's fine. Yeah, that's fine. Yeah. Great. Thanks, Mariela. So on your instrument, you control the sample pressure. So you decide how much pressure you're putting on the cells, on this fluid, to push it through into the center of the sheath fluid. And on your instrument, you can go low, medium, and high. On the caliber, on most of your instruments, you control this. And what you're doing is you're controlling that pump that pushes the air and pushes the pressure on the cells here to push them through. And so for certain things, it's important how much pressure you put. If you're running the cells on low sample pressure, it's got less pressure pushing it. So this is a very narrow core that's going through the center of the samples of the sheath fluid. And so your cells are all positioned in pretty much the same place in the middle of the laser. So you're getting very uniform excitation of your fluorochromes because they're in the middle of the laser where the power is the highest. And so you're getting uniform excitation, which means you're getting very uniform emission. And so for something like DNA cell cycle analysis, it's very important to be running these on low because you want to do very fine measurements of your amount of DNA. If you put the pressure up high, you're pushing it through with a wider core. They're going through really at the same speed, but you've got more cells going through because the core is wider. And so the cells are not necessarily right in the middle of the laser. There's more variation on the excitation and the emission. For most applications, this is just fine. For immunology, this is just fine. And that's what most people will use because they want to get their cells through faster. But if you look at, these are beads running through. If you run the same beads through on low, you can see that you're getting a very tight coefficient of variation around that peak. And if you run the same thing through at high, this is much wider. And so air or bubbles or dirt or clogs on your instrument can also deflect the cells from the center of the laser beam. And so they will get decreased fluorescence, decreased excitation, decreased emission. So it's very important to keep your instrument very clean and no bubbles. And we'll talk about that in the... So we have the cells going through one by one nicely through the middle of the laser. Let's take a look at the optics. So we have excitation optics and we have emission optics. So the excitation optics are the lasers and the lenses and the prisms that focus those laser beams on the point where the cell is going through the flow cell. We use lasers because they're bright, because they're coherent light, they emit at a single wavelength, so it's very clean, they're very stable, they focus to a very tight spot on a tiny area, so you can focus them very tightly. They're getting smaller and cheaper. In the old days when we all started, we were working on the big instrument, and somebody was saying that they were working on a Coulter Epix, which meant that she was aligning the lasers every day, which is not a trivial thing. We did it every day. But now you don't have to do that. The lasers are smaller, they're cheaper, and only your instrument service guy is going to be aligning them for you. The range of colors are now huge, and Bill is one of the big world experts on lasers. and new lasers. He's always testing out the new lasers and putting them on his instrument, and he's pretty much the guru of lasers. And so we saw the lasers on the typical instruments. Let's just look at them again. We've got a 488 nanometer laser that's mostly used for exciting fits in PE. Remember the fits excitation curve? The red laser, the 633, 635 in that range, will excite APC, the APC tandems, Alexa 647. The violet laser is used for all of the new violet dyes, DAPI, be the brilliant violet dyes, Pacific blue, Pacific orange, cyan, all of these new dyes that are coming out. The yellow-green lasers on the new instruments are very useful because we'll see in a minute, or maybe it's Roxana or Florencia that's going to talk about the cross laser excitation. I think it's in the next talk. But some of the, you saw that the excitation curves could also be quite wide. And so something like phycoerythrin excites very well at 488, but it excites even better at 561. And so if you're exciting it to a higher efficiency with another laser, you're going to get more fluorescence. So you're going to get better separation of your positive and negative cells with a laser that really targets the highest emission point. So there's a lot of yellow-green lasers now that are being used for PE, all of the PE tandems, and for the red and fluorescent and cherry proteins. And the UV lasers have generally always been used for something like Perkst or the Indo, but now we've got a lot of the UV dyes and the UV tandems, and so we've got a wide range of fluorochromes that can be used. And now we're up to 35, 40 colors with something with the Aurora. So here's what they look like, the lenses and prisms that direct and focus the laser. So you can see that we've got a bunch of different lasers. We got a green one, we got a red one, we got a blue one, we ut a violet one. And all of these prisms are directing these lasers so that they're coming onto the same trajectory, and they're going to intersect the cell as it's going through the flow cell. And so these lasers are directed so that they will pass in the middle of where the cell is going to go through and at different heights. And we'll see that later. The cells will go through one laser, second laser, third laser, fourth laser. So here we can see all of the different laser beams, and they're all being focused on this flow cell where the cell is going to go through. So the laser beam geometry is has the highest power of a laser beam is in the center. We put beam expanders on them so that they're a little bit wider, so that there's more leeway for the cell going through. But you really want, for the maximum excitation, you want your cell going through the middle of that laser beam. You don't want it going off through the side, because if it's going off to the side where it's not getting as much excitation, the emission is going to be lower. And so when you see that data on your histogram, the cells will be over to the left because they're not as bright, because they didn't get as much excitation. And so that's an important thing with the fluidics. The cells are going through the center of the laser. And you saw even when we put the higher pressure on that there was more variation as it went through. And so this will affect how you see your data. If they don't, the decreased excitation means decreased fluorescence. And dirt or bubbles can cause this by changing the trajectory of the cell path so it's not going through. So everything's being diverted to the side. So keeping, as I said, your instrument very clean will avoid a lot of problems like this. So we've got the cells going through the middle of the laser. The lasers are all directed so that they're hitting the cells as they're going through the middle. They're fluorescing. All of the colors, the different fluorochromes on your cell are all fluorescing in all directions at the same time. We need to take those different fluorescences, separate them, and measure them separately. And so we do this with the collection optics. The collection optics take the mix of different fluorochromes, the emissions from those wavelengths, and separates them out so that they're measured in a separate detector, each one of them. So how do we do that? So the light is collected, as I said, behind. So the laser's coming here, I'm the cell. You've got a lens that's collected here. we'll look at the forward scatter in a few minutes. But so all of the light that's being collected, the side scatter, and all of your fluorescences, say you've got 2, 3, 4 fluorescences, that's all mixed. So you're getting all of the wavelengths from all of the different fluorochromes mixed, collected through a lens there, and then it's directed towards the collection optics. So after it collected by the lens at 90 degrees, it then passes through mirrors and filters, which is going to separate out the different wavelengths that correspond to the emission curve of the fluorochrome that you want to measure and direct it to the correct detector. So we use 3 different kinds of filters for this. We use long pass filters, and these are designated as LP for long pass, and 500 is the wavelength cutoff. So an LP 500 will transmit all of the wavelengths above 500. So if you look here, it won't let through the wavelengths that are below 500, it'll transmit the wavelengths above 500. Similarly, a short pass SP 500 does the opposite. It lets through all the wavelengths below 500. So we have a short pass here. It's letting everything below 500 through. This is the transmission. And we also use bandpass filters. And we'll look at where and how we use these in a few minutes. So a bandpass filter lets through a specific range of wavelengths that's designated by the numbers behind the BP. So a bandpass 530/30, this is how they're typically designated. The 530 is the center wavelength of that filter. So it's going to let through all of the wavelengths that are 530 with a certain window around it. And the window is designated after the slash. So 30 nanometers means that it's going to let through half above and half below. So it's going to let 15 above 530 and 15 below 530. So it's transmitting 530 plus or minus 15. 15 is half of this. So it's going to let through the wavelengths that are 515 to 545. There are a lot of band passes. Every detector has a band pass in front of it, and these are going to refine the wavelengths your detector is seeing so that you're only measuring the wavelengths of the fluorochrome you want to measure. These filters can also be used as dichroics. So it does the same thing. If you angle the filter at the right angle, and the angles differ in the different machines, but you're angling it to use as a dichroic. So all of your lights coming through, this is a long pass LP 500. It's letting through everything above 500, but it's going to reflect everything below 500 in another direction where you can still capture it and use it and still subdivide it. So you're not losing, back. So you're not losing that light, you're just directing it, hitting the wrong button, sorry guys. You're just directing it in another direction. So remember that the emission spectra have tails, and so what we're targeting with the band passes in front of the detector is the highest part of the emission spectra, so that we're really collecting the light from FITC. So 530/30 from FITC is collecting this part of its emission spectra, so you're getting most of it. A 585/42 targeted to collect PE is collecting the highest part of the PE spectrum. And so if we look at a schema, a very simple schema here, but this is the principle that they use. We've got our laser hitting our cell. We've got the lens that's directing it at 90 degrees off to the side here. The first thing that it's hitting is a short pass 560. So the short pass 560 is going to let everything below 560 through, and it's going to reflect everything above 560 in this direction. Over here we have the detector that we want to use for PE. And in front of that detector, we have a band pass that's further going to refine the wavelengths that it sees, because those detectors only see photons of light. You have to make sure that the photons of light they're seeing correspond to the photons of light of the fluorochrome you want to measure. And so you want to make sure it's only seeing the wavelengths of and measuring the wavelengths of those photons of light. And so this bandpass here is making sure that this detector is only seeing the wavelengths that correspond to the highest part of PE. Above here, you've got it everything below 560. The detector for FITC is up here, and here we're letting it see only the wavelengths that correspond to that part of the FITC spectrum, which is 530-30 or 515 to 545. We complicate it a little bit further, so we're looking at the forward scatter and side scatter and adding another color, and this is actually what you see in the caliber. Your laser hitting your cell. Over here, you've got your forward scatter, and in front of the forward scatter detector, you've got a bandpass that's 48810, because light scatter is the same wavelength as your original laser. This is a 488 laser, and so we only want it to measure 488 light. The side scatter and all of the fluorescence is collected here at 90 degrees. First, then hit along past 510. Everything above 510 goes here. Everything below 510 goes over here. This is the side scatter detector. And so here it's also measuring side scattered light at 488 nanometers. Now we're separating out the fluorescences here. LP 560, letting everything above 560 through, directing everything below 560 to the FITC detector with its bandpass. Up here along past 610, going above, letting everything above 610 through, and directing everything below 610, so that's between 560 and 610, over here to the PE detector with its bandpass, which is refining those wavelengths. And then up here we've got along past 650. That means that this detector here is seeing everything above 650, which means that if you've got PE Psi 5, PE Psi 7, per CP up here, it's going to detect all of those. So here's a little schema. And so on the different instruments, these optical filters and bandpasses are used in slightly different configurations, but they all use those same principles. So here we've got a linear array. I think this might be a Galios. And so your light is coming in here. It's hitting a 550 short pass first, letting through everything below 550 to the FITC, which is targeting around the 525. Everything above 550 is going over here, PE, and so it's bouncing the light back and forth. These are the BD instruments. So this is what you've got on your area. The light is coming from the flow cell through a fiber optic and bringing it to an octagon. And so this octagon, the first thing it hits here is a 735 long pass. So it's taking off the higher wavelengths first. Letting through everything above 735 here, there's a band pass in front of this detector. This is PMT number A. And that's detecting APC Psi 7, PE Psi 7. 780-60, it's letting through only 780, 750 to 810. Everything below 735 is now bounced over here to the 655 long pass, lets through the next layer of highest ones. There's a band pass in front of the B detector, bounced over here, bounced over here, bounced over here, and at the very end, you're taking the lowest wavelengths, which happen to be the side scatter. So far, we've only looked at a single laser. We haven't looked at multiple lasers. Remember, I said that they were focused onto the stream. So what happens when you have more than one laser? The lasers are separated in space from the alignment, and so they're also separated in time. The cell's not going, it's going to go through one laser, then another, then another. And so we have our cell here coming down. The first thing it's going to hit here is the 488 laser. So the fluorochromes on that cell that are excited at 488 are going to fluoresce. And that emission is going to be collected at that point in time by the detectors. It's continuing down. And then it hits the next laser. And so the fluorochromes that are excited by 633, so that would be APC, APC Psi 7, Alexa 700, those are going to be excited and fluoresce. And they're going to be, that light is going to be separated and collected by the detectors. And so since it's separated in time, in space, it's also separated in time. So it takes a certain fixed amount of time that cell is traveling to get from the first laser to the second laser. And the electronics on the instrument know how long that's going to be. So they know that that cell, when it hits the second laser, is the same cell that went through the first laser at a certain time, so it can correlate the fluorescence from the different lasers into a file cell by cell, so it has the information for the same cell from laser 1, laser 2, laser 3, laser 4, and that goes into your data file. And we'll look at the data files in a second. And so this laser time delay is very important, and there's a different specific time delay on each instrument for the amount of time it takes for that cell to go from laser 1 to laser 2 to laser 3 to laser 4. And here's, I think this is an area, you can see the pinholes behind where this light is collected, and then there's a fiber optic that's taking those emissions from that cell as it goes through each laser. It's taking it to the specific octagon or trigon detection system that's going to separate the light from the fluorochromes that are excited by that laser. And so it's going to go through here, laser 1. So this looks like a violet laser. So this light here is going through a fiber optic. It's going to the detector for the violet. So it's going to separate out your brilliant violet 421, your brilliant violet 615. It's going to separate out all of the fluorochromes for that cell that were measured. Then it's going through to the blue laser, and then it's going to, so that light will go to the detectors for the blue laser. This will go to the detectors for the green laser. And so there'll be a time difference that's correlated. And so when you get your final data file, it's very important that the time delays are correct so that it's correctly assigning the fluorescence to the right cell. And so this is what it looks like in reality. So you've got, this is a caliber. You've got your blue laser that's coming through here. Here's your red laser. It's being directed over here. Your cells are coming up here. Here's your flow cell. Your lens behind is sending it back here. This is your forward scatter detector. And then here are your diachroic mirrors that are sending it in different directions. These are your photomultiplier tubes that are detecting the photons of light. And in front of each one of them, these little metal things, those are your bandpass filters. And so I'm not going into the full spectrum because Maria, lucky to have one of the world experts on spectral flow cytometry, she's going to be teaching you that, and she'll do a much better job than me. But in general, with a full spectrum, you're measuring the full spectrum, not just certain bandpass filters. You're measuring a series of light all the way across for each laser, which gives you a lot more information. And I'm going to skip this and let Maria do that. And so now we've got the optics and fluidics. Now we'll take a look at the electronics. So the electronics overview is that you've got your photons of light that are coming from your cells. They're going through this last bandpass filter. So this detector is only seeing a certain range of wavelengths that corresponds to the fluorochrome that you're hoping to measure in that detector. And so these light detectors are going to transform light photons into electrical current. They're going to emit a voltage pulse that's proportional to the amount of photons and proportional amount of emission that came off of the cell at the flow cell. Then this voltage pulse is going to be digitized to a numerical value. So a smaller voltage pulse will have a lower numerical value, and these are channel numbers. And so your data file is going to give you a series of channel numbers that's going to correspond to the intensity of the fluorescence that came off that cell. And so let's take a look at the detectors and how they work. So the photodetectors transform light into electrical current. The type of photodetectors that we use in cytometers are basically photodiodes and photomultiplier tubes. The photodiodes classically were used for forward scatter because they weren't very sensitive, and forward scatter is a very strong signal, and it would burn out a very sensitive detector. So on the older instruments, this is usually a photodiode. There are new avalanche photodiodes now that are used in a lot of instruments. The Cytoflex, the Aurora uses photodiodes. They're avalanche photodiodes. They're very sensitive, and they're particularly sensitive for the red wavelengths, which don't have a lot of energy. And the photomultiplier tubes are used for the weak signals. So the side scatter and all the fluorescence parameters are generally measured using a photomultiplier tube. And this is what a photomultiplier tube looks like. Remember, we saw the diagram in the 3D picture of the thing. So it's got a photosensitive area. So here's your photomultiplier tube, and it's plugged in there. And your light is coming this way. You've got your filter here that's only letting through the photons of the light you want. And this is your photosensitive area. This is your photocathode. And so these light photons are hitting this cathode, which is then emitting an electron for each photon. And then it goes through a multiplication system. There's a dynode chain which multiplies these electrons so that at the end of this multiplication system, you get a lot more electrons than left the cathode. And that will increase the sensitivity of the measurement. It will also increase the background. So you have to find, and we'll talk about it in the labs, you have to find the right settings for the multiplication system. But it's going to, at the end, give you a voltage pulse that's going to correspond to the number of photons that hit this photocathode. And here's another one. And so on your instruments, most instruments, there are a few instruments that don't yet let you change the multiplication system on your PMT, but most of them do. This is DIVA, this is a BD instrument. And so for each PMT, each detector, you control the multiplication on this to make more or less electrons come out at the end for each electron that left the beginning. So you can increase the sensitivity of the signal. Remember, you're also increasing the background when you do that. And so that you'll get a bigger or a smaller pulse coming out, depending on what you put here as the setting for the multiplication system here. So the signal that you're getting out of that is going to depend on the original number of photons that were emitted by the fluorochrome, plus the multiplication system that you control. And we'll look at how to do this in the lab. And so you can have like 10 to the 3 to 10 to the 8th electrons to get to the anode for every electron that left the cathode, depending on the voltage that you put here. So how do you get this voltage pulse? So what happens, the cell is coming up through here. You got your fluorochromes, these little pointy things on your cell. It goes into the laser. As it goes into the laser, it starts emitting fluorescence. The fluorescence starts getting through the filters, gets to the photomultiplier tube. The photomultiplier tube then starts emitting a voltage pulse. And so the cell will go into the center where it's maximally excited, and then it goes out to the other side and the voltage pulse goes down. And so here you can see the cell going in, time, the top of the voltage pulse, and it comes back down again. So as it goes through, over time here, you've got time and the voltage on the y-axis. The cell goes in, you get a maximum, and it goes back down again. And so you can measure a number of things off of this voltage pulse. The signal processing will calculate these for you. And so you have the height of the voltage pulse, and on the instrument, quite often it's signaled as FITS H, FL1 H. So H afterwards, it's giving you the measurement of how high that voltage pulse, which is the maximum of the fluorescence when the cell was in the middle of the laser. It will give you the area, which is what is mostly used, is the area of the under the pulse from the time it entered the beam to the time it went out. So you've got all the cumulative fluorescence. And it also gives you the width. So it'll be FL1 width, forward scatter width. That's the amount of time in microseconds that the cell was within the laser beam. And this is useful, the height and the width are useful for discriminating aggregates. And Bill will talk about that in the cell cycle. We also use it generally for excluding doublets, but he'll show you a schema, or I can do that in the lab. This afternoon I'll do a drawing. And so you've got a series of pulses going through here. You've got some cells that don't have much fluorescence. You have cells that have a lot of fluorescence. And you're going to get a channel number, which is your signal intensity. which is the digitalized number that the voltage pulse give you. So a low voltage pulse is going to give you a low channel number, and a high voltage pulse, if you've got a lot of fluorochromes, is going to give you. So in a perfect world, they'd all be in exactly the same channel because the measurements would be absolutely perfect. But in actuality, you get a Gaussian around the curve. And as we saw earlier, depending on how much pressure you put on for the cell going through, if you put low, medium, or high, you're going to get a narrower peak or a wider peak, which may not matter. In this case, it's separated by logs. But if you're looking at DNA cell cycle analysis on a linear scale, you're going to want them as tight as possible because you want to know if there's any little aneuploid populations that are nearby. And so a smaller pulse, less fluorescence, more fluorescence, and you're going to get bigger pulses. And so this is digitalization. The pulse size numerical values, the channel numbers are recorded. The data is saved as a list mode file. So for every detector, for each cell that goes through the laser, you have a voltage pulse that's digitalized for every single detector. It's processed, and then it's collected in a list mode file. And basically, it's cell by cell, and you have the value of the fluorescence for each of the fluorochromes correlated to that cell. And so you can look at these a number of different ways. You can either look at how many cells have how much fluorescence. So this is just intensity of fluorescence here and number of cells. So you can see here that there's a fairly wide peak here of low intensity or negative cells, a very high peak of high intensity positive cells. Or you can look at two different colors, two different fluorochromes against each other. So you could look at green versus red. And in that case, you see that you've got three populations. You've got a negative, you've got one that's slightly positive for this, and then you've got one that's positive for this and negative for that. We'll look at that a little bit more. And so this is what you get. You get your cells in sequence, and you have all of the values measured cell by cell for all of the fluorochromes that you're measuring and the scattered measurements. So this is what it's going to look like, and you're going to have a list mode file, your list mode file. So here you're looking at FL1, which on the older machines was always FITSI. This here over here is APC. You've got low and you've got high intensity. Let's get through this. We just looked at that. So now we can answer the questions. How many big cells? How many little cells? And of interest is the fact that we can gate on the cells. So if we're looking at the small cells here, or the monocytes or the granulocytes, we can set a gate around those, and we say, I only want to look at the lymphocytes, and then we can look at the fluorescence of those particular cells. And so here we're looking at a single parameter histogram. We're looking at the fluorescence intensity of red, which would be APC, and it looks like we've got two populations, low intensity and a high intensity population. Whereas down here, if we look at the red here on the y-axis, and we look at it against green, which is giving us another antigen that we're measuring, we can see that where we had a low and a high here, in actuality, we have three populations. We've got a negative population, but the positive population has some cells that are negative for green and some cells that are positive for green, so they're double positives. And so this is the value of being able to look at all of these. You can look at anything against anything, and you can look at any subpopulation that you want. So we could do the same thing and say, okay, I only want to look at the granulocytes, or I only want to look at the monocytes. Changing the PMT voltage. How am I doing for time? 10 minutes. Okay, that's okay. So we're looking at one cell, the same cell here, going through the laser, but we're changing the voltage that we're putting onto that detector. This is forward scatter, so we're running it through once where we put it at 300 volts. So we're not multiplying those electrons very much, so that voltage pulse is going to be small coming out of that detector. If we push our voltage up to 400, the same amount of photons are hitting, the same amount of electrons are leaving the cathode, but we're multiplying those electrons. So at the end, the voltage pulse is bigger and it's digitalized to a higher number. So you can see that this is all relative. If we pump this up to 500, the same amount of photons, the same amount of electrons, But now at the end, at the anode, we're getting a much bigger number of electrons, a higher pulse. And so it's out here on the right-hand side. And so we can change the position here depending on the voltage that we put. We also need to tell the cytometer what is a cell, what isn't a cell, what we want to measure, what we don't want to measure. And because it has to have a threshold. You can't measure just background. And so every cytometer needs a threshold to determine what is a cell, what is an event, what is an extracellular vesicle, and what is background or debris, which will be excluded from your analysis and will be lost in your data file. So the threshold is the level above which detected signals will be processed and saved. If a pulse is lower than the threshold, it won't be seen and it won't be saved. It'll be excluded. So if you think you see it running through your cytometer when you're acquiring and you don't have your settings right, when you go to look at it a week later, your cells may not be there. So it's very important how you set your PMT voltages and how you set your thresholds. And so if we look at these same cells here, let me skip back here. Whoops, going the wrong way, sorry. So we've got the same cell. We're doing the same thing here, and we've set a threshold. And now we're looking here at a 2-parameter. We're looking at forward scatter versus side scatter. We're running the same cell through at 300, 400, to 500. And here we've put a threshold. So we've set the threshold, and you have to set a threshold. There's always a threshold, whether you set it or not. The threshold is here. This cell is being shown here, but you won't see it because it's below threshold. It won't appear. If you push that voltage up to 400, now it's above threshold. You will see it. And if you push it way up to 500, it's going to be over here, like we saw it. So where it's important is in something like this. We've got a mixed bag of cells going through. We're running these through at the same PMT voltage. So I'm not changing the PMT voltage here. I'm only changing the threshold. But because you've got bigger cells and smaller cells, you've got bigger pulses and you've got smaller pulses. So the big cells are these big ones, big voltage pulses, and the tiny ones are here. If I set the threshold very low, you can't even see it here, but it's there. I can see the very tiny ones. I can see the lymphocytes, the monocytes, the granulocytes. I can see everything here. And I've got a whole big population here that are these little guys. And they're above threshold, so they're showing. If I put the threshold up, and you do this with the software, so I'm pumping the threshold up, saying I'm putting it at 10,000 instead of 5,000. And these little guys now are below threshold. And so now you can actually see where the line of the threshold is. And you can see that there are things being excluded because you can see that there's stuff that's on the axis, that's on the edge. And so you know that you're missing some stuff here. Maybe you want it, maybe you don't want it. That's a decision you have to make. But you need to be aware of what's there and what's not there, because you want to be sure that the cells that you want to measure are above threshold, because otherwise they're gone for analysis. And so here you can see this is okay. We've lost half of this. We know that there's probably a population here because we can see all this stuff here. We've got our lymphocytes, monocytes, granulocytes. If I pump it up even higher, say to 20,000, I'm losing some of these middle-sized ones. And the threshold is up here somewhere, there probably, but I don't see anything at all there. And if I ran it like that, I wouldn't know that those were there. And so it's really important how you set your PMT voltage and how you set your threshold. And we're going to go through that in the lab this afternoon. How do you set that? Because you probably don't want these. These are probably red blood cells, or they might be platelets, or they might be debris. But you need to know that the cells that you're interested in are going to be in your data file. And here's your laser time delay. Remember, I talked about how it correlates. So here your cell is going through your first laser, which happens to be the blue laser here. 13 microseconds later, it's going through the red laser. So those measurements are going to be made there. And 22 microseconds later, it's going through a violet laser. And so if you look at this, here's time in this direction. You're getting all your pulses in the detectors that are measuring off your blue laser. Here you're getting the measurements that are coming off your red laser, 13 seconds, microseconds later. And then 22 microseconds later, it's getting excited by the violet laser. Now, these windows that are here are your windows for the pulse measurement. So your instrument is saying, okay, I know that cell is supposed to be going through at 13 microseconds. I'm going to measure at this little window around 13 microseconds. And I'm going to measure at this little window around 22 microseconds. And so it's going to digitalize this pulse at that time. If you have a clog or something, and they're going through slower than they're supposed to, then this one's going to be going through here. It's going to be outside the window measurement. And so, again, you need to keep your instrument clean. You don't usually determine the laser delays. Those are usually determined automatically by the instrument. So we've got some references here. And, oops. These PowerPoints will all be up on the website for the things. And I also have a chapter that we wrote that's in current protocols in immunology for 2017. All of this is in there. And there's a lot of good stuff on the web. Good basic tutorials. And gracias.
Gracias, Zosia. Muy bueno. ¿Preguntas? A ver, ¿se animan? ¿Alguna pregunta? Sí.
Hi, I will
Hi.
try to speak.
Yeah, and I don't hear so well, so you have to speak loudly.
When you're setting your threshold,
Yeah.
in the cytometer, you put a number, right?
Yeah.
Like 10,000.
Yeah, yeah. Sometimes you have a slider, sometimes you put the number in. Yeah.
Never understood how to correlate that number with the population that I don't want to acquire.
Okay, so it's a channel number. So on your scale that's going like, say on a BD machine, 252,000 channels, it's going to be at 10,000, 20,000. So you're determining what channel it is. Where your cells are is another question. And if you don't know, it's the first time you're doing it, you don't know, have no idea where your cells are. Usually it's a good idea to have a marker. So say you know that the cells that you want are CD4 positive. So you set a very low threshold. You set your voltage for your forward scatter very high because that's usually your trigger threshold. You can put your threshold on other parameters. Sometimes with cell cycle, it's done on the DNA dye. But in general, most people put their threshold on forward scatter. So you want to be sure everything is above. And then you look at your CD4. You look at your positive CD4s and you set a gate around it. And then you gate back to that forward scatter, side scatter, and you look at where your CD4 positive cells are. If they're off the top of the scale, bring your PMT voltage down. If they're at the edge of the bottom, and you can see that some of them are cut off, you're going to have to move that PMT up or move your threshold down because you want to be sure that the whole cloud of that cell is above threshold. And so that's a good way to find cells. I have a QC and troubleshooting talk that I can load up as well. I'm not giving it here, but we can talk about all this stuff in the lab. But sometimes when you're working with stuff like solid tumors, where you've got huge amounts of dead cells, even, you know, we do this all the time, and even we have trouble finding the cells. Where are the live cells in all of this debris? A dead cell marker is really helpful. So what you can do is you have your cells put in a dead cell marker like DAPI, and I start out by putting forward scatter against DAPI on log, and I move the PMT voltage up so that I'm sure that the very positive cells for DAPI, and it's like a DNA histogram because they're very positive, make sure that those are probably naked nuclei, but at least you know that if you've got them above threshold, your live cells are probably bigger and you have those. And then you can quite nicely see the dead cells and the live cells and where to gate them. And so in the QC and troubleshooting one that I've got, there's a description of that, and there's some slides on that. So you can take a look on how to do that. It's really hard at the beginning to find your cells. Once you've figured it out, then the next time you go back, you've got a template, you've got your settings, you just do it over and over again. But it's not always easy. We'll talk about setting your PMT voltages because how you set those is important too. And it's mostly important because it's specific for the instrument and the detector, not necessarily for the cells. So once you know what your best voltage is, you pretty much start with that every time. The only time you need to change that is if you've got positive cells off the top of the scale. You don't want your positive cells off the top of the scale, but so there are ways, filtrations to do that, and we're going to do that in the lab. And there was something else. Oh yeah, and the troubleshooting thing, it's like, and this happens all the time. You know, your core facility people, you call them at 10 o'clock at night, they're not too happy because you can't find your cells. But there are steps you can do, and running QC beads is absolutely essential to knowing whether your instrument is working and finding your cells. If you're sitting in front of the instrument and nothing's going by, you put your QC beads on. You make sure that those are where they're supposed to be. If they're not, you have to get the instrument working. It's a problem with the instrument. If they're good, make sure you got cells in your tube. You know, look at a microscope. A lot of times people put in 10,000 cells and then they wash them, they fix them, they permealize, and there's nothing left. Make sure there's something there. And then if they are good, the next problem is threshold and PMT. They're below threshold somewhere.
Okay, thank you.
So the steps are, and the QC, running the QC every day and running the QC whenever you have a problem, having an easy one to set up on your instrument is really important. That's your key.
Thank you, Zosia. And also, it, as Zosia mentioned, depends on the cells, the size of your cell that you are looking for, because normally in a core facility, you had the threshold as the fall, more or less, and suddenly once a user is bringing mammalian cell, and then later on someone with bacterial cells. Then it's not the problem. And even now, with extracellular vesicles that is below this threshold, so it's another issue. So that's these important things that Zosia was mentioning, that first control your equipment, then just with known samples like you see, comparla con esas cosas. And then the other issue is that if you are wanting to concentrate on live cells or whole population, for example, when you search for apoptosis, so sometimes you set up the threshold too high in a way that you don't want to have dead cells. But if you are analyzing apoptosis, in this case, you have to go down with the threshold. So Bill is expert in apoptosis and cell cycle, and I think you are going to deliver a talk also on cell cycle. And on Friday, vamos a tener una charla de troubleshooting. Así que ahí troubleshooting always are welcome. Troubleshooting during the course and troubleshooting during the daily life. So I don't know. I think
you can ask us, you know, if we don't know the answer, we usually know somebody who does. And it's hard, like if you can come up to a side to sit down in front of the cytometer, and the settings that the person has put on before are completely different from what you need. And sometimes it's hard. But you'll get used to it. You'll find it.
Okay. Thank you very much. Gracias. Time for a coffee break. And we will meet in half an hour. Yes.
Is that okay there? Yeah. Oh, I think sure.
Bueno, vamos a dar continuación a las clases. Antes de comenzar, les voy a pedir que lo que les comentaba antes, en algún momento o cuando volvemos del almuerzo, vamos a pasar a presentarnos o dicen su nombre, su lugar de trabajo y qué es lo que están haciendo, si están haciendo un posgrado o son técnicos o son investigadores, investigadoras. Si tienen conocimiento básico, intermedio o avanzado de citometría. Y después le vamos a repartir a cada uno, o de la libretita que tienen, sacan un papel y ahí anotan máximo 3 objetivos que ustedes esperan del workshop. Lo vamos a anotar hoy, el nombre y los objetivos. Y lo vamos a poner, si quieren lo podemos después luego pegar acá y después al final del curso, de nuevo, vemos si se cumplieron esos objetivos o algunos sí o parcial y así, ya sabiendo de entrada cuáles son los objetivos de cada uno y la formación también uno puede acomodar un poco las clases. La otra cosa que les decía es, el curso va a ser en 2 idiomas. Tenemos speaker que van a hablar en inglés, que es Zosia, Bill y Sophie Nouveau, virtualmente de Pasteur París. Y también tenemos docentes de lujo, de habla hispana. Una de ellas es Florencia, del Río de la Plata, de Argentina. Rosana, que es de México, pero que está trabajando en Estados Unidos. Mañana vamos a tener a María Jaimes, que es colombiana, que está trabajando en Estados Unidos. Y Lola, que es de España. Así que, como les decía, aprovechemos esta instancia de toda esta semana de interacción y de sacarse las dudas y no ser tímidos o tímidas para hacer preguntas. Entonces ahora, con esto, dicho esto, voy a pasar a presentar a Florencia Quiroga. Bueno, Florencia se graduó de la Facultad de Farmacia y Bioquímica de la Universidad de Buenos Aires, donde recibió el título de Bioquímica y luego el doctorado en Inmunología. Realizó su formación de postdoctorado en la Facultad de Medicina de la UBA, investigando la regulación de la respuesta de las células T durante la infección por Mycobacterium tuberculosis. Actualmente es investigadora independiente del CONICET y docente de la Facultad de Medicina de la UBA. Desde el 2008 dirige su propio grupo de investigación, que integra las áreas de experiencia obtenidas durante sus estudios de doctorado y postdoctorado, centrándose en la respuesta inmunitaria antituberculosa innata y adaptativa en el contexto de la infección por HIV. Tiene más de 40 trabajos publicados en revistas arbitradas en esta temática. También integra el Consejo Asesor del Sistema Nacional de Cytometría de Flujo de Argentina, que depende del Ministerio de Ciencia y Tecnología e Innovación Productiva, y también integra el Consejo Directivo del Instituto de Investigaciones Biomédicas en Retrovirus, SIDA, INVIRIS. Es una gran docente con un gran compromiso por la educación, lo que se ve reflejado por la enorme cantidad de cursos que ha realizado sobre educación y pedagogía. Y bueno, es un placer para nosotros tenerte aquí, Florencia, y bueno, bienvenida.
Bueno, muchísimas gracias por la invitación. ¿Se me escucha? Bárbaro. Muchísimas gracias por la invitación a este curso. La verdad que es un placer poder brindar la experiencia que nosotros tenemos en la facultad, acumulada en todos estos años, y bueno, esperando que el aporte sea fructífero para todos ustedes, ¿no? Finalmente los que están asistiendo. Así que mil gracias, Mariela, por la convocatoria, por la invitación, y bueno, demos comienzo. En esta instancia, bueno, lo que les voy a pedir es que cualquier duda que tengan, cualquier comentario que quieran hacer desde su experiencia, lo hagamos. Van a ver que yo siempre voy a estarme echando la tarea de investigación que haga. Yo soy investigadora y hago citometría de flujo desde que soy así de chiquitita, entonces es parte de mi vida, es una pasión que tengo, y por eso devine en Core Manager, por decirlo de alguna manera, en el laboratorio donde trabajo. Pero siempre tomo la citometría de flujo desde el punto de vista de responder preguntas, integrarla a un proyecto de investigación. Entonces está buenísimo que cada uno desde su lugar ponga su visión porque hace crecer al resto, ¿no? Así que bueno, empecemos con esto. A ver. Bueno, estos son los objetivos que delineé para esta clase, que como tengo alma de docente, siempre los quiero delinear, y al final de toda esta charla me gustaría que ya un poquito lo vimos con Zosia, de qué se trata el mecanismo de fluorescencia, cuáles son los compuestos fluorescentes más importantes que existen, que se utilizan en citometría de flujo, cómo funcionan los fluorocromos en tándem, y que analicemos un poquitito en detalle de qué se trata el solapamiento espectral, cuáles son las consecuencias en la emisión y en la detección de fluorescencia, y qué problemas nos generan y cómo resolverlos, ¿no? Entonces, a partir de ahí, conocer el fenómeno de derrame espectral, incorporar conceptos involucrados en las herramientas disponibles para corregir ese solapamiento espectral, todo lo que es matriz de compensación, de derrame espectral y controles FMO, no se preocupen porque lo vamos a ir viendo detalladamente a lo largo de la charla. Y por último, muy chiquito, pero también como para introducirlos de a poquito, que conozcan la configuración de los detectores, que ya la vimos con Zosia, pero conocer la configuración de los detectores convencionales y también compararlo con la de los, de los citómetros espectrales, ¿sí? Como para ir viendo diferencias entre ambos tipos de equipos. Ahí va. ¿De qué se trata la fluorescencia? Ya un poquito lo vimos con Zosia. Nosotros tenemos un compuesto que, como ustedes vieron, son compuestos que tienen electrones con capacidad de ser excitados, ¿sí? Anillos aromáticos, ese tipo de cosas. Viendo atrás, lejos en la facultad, esas moléculas que tienen las nubes de electrones con movilidad, como son los anillos aromáticos, bueno, que reciben energía. Esa energía va a ser recibida a partir de una fuente de luz, ¿sí? En nuestro caso va a ser una luz láser. Por lo tanto, esta luz va a ser absorbida y va a llevar a los electrones desde su lugar basal hacia una situación de excitación, ¿sí? Esa absorción de energía entonces va a llevar al electrón a una situación de máxima energía, va a haber pérdidas transicionales mínimas de energía y una última pérdida grande de energía por parte de ese electrón queriendo volver a su estado basal, porque nosotros siempre queremos volver a un sistema de entropía, emitiendo luz. Esa luz es de menor energía, como les dijo Zosia, por ende de mayor longitud de onda. Entonces, ese sería el proceso de emisión de luz por parte de un fluoróforo en términos bastante rudimentarios. Ahora bien, este proceso lo pueden generar diversos tipos de moléculas que nosotros vamos a llamar fluorocromos, porque van a ser aquellos que emiten fluorescencia. Y en citometría de flujo tenemos diferentes tipos de fluorocromos a los cuales nosotros utilizamos y les sacamos el mayor provecho. Los más conocidos son los orgánicos, los primeros conocidos, como la fluoresceína o la ficoeritrina, fíjense en su estructura, siempre con esos electrones disponibles para ser excitados. También los biológicos, como la ficoeritrina o la aloficocianina, que son moléculas grandes, proteicas, ¿sí? Bien voluminosas, que tienen un efecto, ¿no? Quizá son más difíciles o un poquito más problemáticas al momento de hacer marcaciones nucleares, por ejemplo. Pero que tienen una muy buena eficiencia en cuanto a emisión de fluorescencia. Después tenemos las moléculas en tándem, que las vamos a ver ahora un poquito más en profundidad en la próxima diapo, que se trata de conjugar dos compuestos fluorescentes, ¿sí? Dos moléculas fluorescentes. Los polímeros orgánicos biconjugados son los más nuevitos y que han avanzado desde que salieron de una manera monumental. ¿Por qué? Porque son muy estables, son muy eficientes para emitir luz fluorescente, y además son muy versátiles, en el sentido de que un pequeño cambio en la molécula induce un cambio importante en la luz emitida, en la capacidad, en la longitud de onda de la luz emitida. Entonces se pueden obtener diversas moléculas con emisiones diferentes, ¿sí? Siempre con la excitación del mismo láser, con lo cual son moléculas muy eficientes y además con cierta facilidad para conjugar a proteínas, lo que los hace ventajosos con respecto a estos últimos nanotecnológicos, los quantum dots, que cuando salieron sonaban perfectos, porque divinos, vos podías manejar según el tamaño de la partícula, la longitud de onda de emisión, con una emisión, un espectro de emisión súper angosto, entonces eran fantásticos porque no había solapamiento espectral, pero bueno, el problemita que tienen los quantum dots, y se refleja en el mercado, es que no son muy amigables al momento de conjugar a proteínas, con lo cual, o por lo menos eso es lo que leí. Con lo cual, y lo muestra cuando uno mira para comprar un anticuerpo, ve que no hay mucho quantum dot dando vuelta para citometría, con lo cual asumo que esa será la razón. Volviendo entonces a las moléculas en tándem, ¿cómo funcionan? Nosotros dijimos que un fluorocromo recibe la luz láser, se excita hacia un lugar de mayor excitación, pierde un poco de energía, pero la mayoría la pierde en su salto a su estado basal y emite una luz fluorescente. Bueno, en el caso de los tándem, el tándem es una conjugación entre dos fluoróforos, ¿sí? Una unión covalente entre dos fluoróforos, a eso me refiero con conjugación. Entonces, el fluoróforo en tándem lo que hace es recibir la luz emitida por el fluoróforo principal, con lo cual tiene que tener un espectro de absorción que acepte la emisión del que tiene al lado, ¿no? Obviamente. Entonces ocurre lo mismo, pero siempre con una menor energía. El electrón basal del tándem se excita y emite en fluorescencia. Esta fluorescencia emitida va a ser de menor energía aún, con lo cual la longitud de emisión va a estar más alejada aún que la emisión del primer fluorocromo. ¿Se entiende? Fantástico. Cualquier cosa me preguntan, ¿no? Perfecto, perfecto. Mariela me hace la aclaración para los que están viendo por YouTube, que por favor pongan sus preguntas en el chat. Está bien, ¿no? En el chat. Que las chicas lo ven en vivo y las transmiten acá en el salón. Así que tanto los que están en el salón como los que están viendo la transmisión, por favor, si quieren hacer alguna pregunta, háganla, o algún comentario o alguna acotación, que la reproducimos acá. Así que bueno, el fluorocromo en tándem nos genera nos abre una ventana nueva en cuanto a posibilidades de generación de fluorocromos. Pero ojo, porque esto son dos moléculas unidas covalentemente. Entonces, puede haber situaciones en el ambiente donde está esa molécula que puedan dañarla de alguna manera y desacoplar estas dos moléculas. ¿Qué pasa si se desacoplan estas dos moléculas? Empieza, porque el que está pegado a la proteína es el fluorocromo principal. Entonces, pierdo el tándem B, me queda el fluorocromo A, y ese es el que va a emitir. Con lo cual, si además en esa célula yo estoy marcando con un anticuerpo que tiene el fluorocromo A peladito, no voy a poder distinguir ahora lo que es el marcador con fluorocromo A y lo que es el marcador con el fluorocromo en tándem. ¿Lo ven? Así que cuidado con las situaciones de desacople. De repente no funciona. Y por eso es que les hice esta listita de consideraciones con respecto a los tándem. Y se trata de, una vez que hacemos la marcación, adquirir las muestras en el equipo lo más rápido posible. Hay veces que uno trabaja en un instituto y tiene que trasladarse para llevar sus muestras a otro. Bueno, este tipo de cosas hay que tenerlas en cuenta y tiene que fijar sus muestras. Mantener las muestras al abrigo de la luz, minimizar el uso de fijadores con paraformaldehído. Hay algunos que son comerciales que no utilizan tanto paraformaldehído, entonces no son tan dañinos. Pero bueno, si el tándem se degrada y se desacopla, nosotros tenemos manera de verlo, ¿sí? Por ejemplo, si hacemos la compensación automática, los valores de superposición espectral van a ser aún mayores al 100%. Ahora, cuando yo les explique de qué se trata la compensación, van a comprender por qué estos valores van a ser mayores al 100%, pero un poco ya lo intuimos, ¿no? Tenemos un fluorocromo A que emite en su longitud de onda y un tándem que es A más B que emite en otra. Si el tándem pierde su conjugado, van a emitir los dos en la misma longitud de onda y eso me genera un problema. A ver, ahí va. Bueno, haciendo todo un raconto de los diversos tipos de fluorocromos que existen y la versatilidad de algunos de ellos para absorber y emitir la luz, vemos que hay todo un espectro de moléculas que pueden ser utilizadas para conjugar a proteínas, anticuerpos o lo que uno necesite para la citometría de flujo, donde las líneas punteadas corresponden a los espectros de absorción y las pancitas llenas corresponden a los espectros de emisión de cada una de estas moléculas. Las líneas corresponden a las líneas principales de los láseres que nosotros utilizamos. Acá está el láser violeta, acá está el láser azul, acá está el green, el yellow green, el rojo, ¿sí? Entonces, de acá podemos sacar varias conclusiones. Primero que podemos utilizar millones de combinaciones. Pero además, fíjense lo que decía Zosia, ¿no? Los espectros de absorción. Si buscamos, tenemos moléculas que pueden recibir la excitación, por ejemplo esta, que pueden recibir la excitación de dos láseres. Obviamente va a haber una línea principal de absorción de cada fluorocromo, pero va a haber líneas secundarias también de absorción, y esas corresponden a otros láseres. Entonces todas esas cosas las tenemos que tener en cuenta, y ahora las vamos a charlar un poquito más, para hacer la elección correcta de fluorocromos en un panel que yo quiera construir, ¿sí? Lo mismo para que observemos las características de los espectros de emisión. Son no son una línea. Si uno piensa en ese electrón que se excita, que pierde energía en varios pasos y que después se emite, esa pérdida de energía en varios pasos no es siempre la misma. ¿Bien? Lo mismo la excitación inicial, como Zosia les contó, que dependiendo del punto del láser que esté tocando a la célula, si el centro tiene mayor energía, los lados tienen menos, la excitación también va a ser menor. Entonces, bueno, todas estas situaciones hacen que la emisión sea un espectro, sea un rango. Voy a tener una emisión mayor, pero también voy a tener colas. Esas colas me van a generar problemas que voy a tener que resolver para mi experimento. Ese problema se llama solapamiento. ¿Bien? Entonces, este solapamiento espectral es el factor más importante que afecta nuestra resolución y sensibilidad en los experimentos de citometría de flujo cuando hago un panel multicolor. ¿Bien? Y se trata de el derrame de fluorescencia en otros canales. Si nosotros vemos los espectros de emisión de estas tres moléculas, fluoresceína, ficoritrina y aloficocianina, vemos que hay solapamiento en los espectros de emisión. ¿Bien? Este solapamiento en los espectros de emisión va a ser detectado por los detectores de los diferentes fluorocromos. Esto va a contribuir al background y a la sensibilidad de resolución en esos canales. Pero bueno, ese solapamiento lo podemos corregir y se corrige en un proceso que se llama compensación. Acá lo tenemos más en detalle, los espectros de emisión de fluoresceína y ficoritrina, y vemos, y las dos barritas son las bandas de detección. ¿Bien? Son las longitudes de onda que son captadas por los PMTs, por los detectores. ¿Ok? Entonces, vemos que el detector de FITC ve solamente la emisión, la fluorescencia emitida por FITC. ¿Eso lo ven claramente? Bien. Pero el detector de PE ve una porción de fluorescencia que le da el fluorocromo FITC. Bien. Esto es lo que nosotros llamamos solapamiento espectral. Cuando un fluorocromo en su espectro de emisión, en su cola de emisión, aporta al detector que no le corresponde, digamos, ¿sí? Entonces, esto nosotros lo tenemos que corregir. Esta diapo la saqué de, esta figurita la saqué de AppCam y me gustó porque básicamente nos habla de lo mismo, pero nos habla de cómo corregirlo también. Entonces, si no aplicamos la compensación, la cola de emisión de FITC, fíjense que estoy con el mismo gráfico, tiene diferentes colorcitos, pero estoy en la misma. Tengo la emisión de FITC, la emisión de P y las dos barritas de los detectores. Y acá me muestra la porción de fluorescencia en FITC que es detectada por el detector de PE. ¿Bien? ¿Lo ven todos? Fantástico. Yo en este momento estoy corriendo células que las marqué solo para FITC. No hay PE. Usé un anticuerpo que estaba conjugado con FITC y marqué mis células. Y las corrí. Y veo esto. ¿Esto es real? No, no hay PE. No debería haber nada acá. Bueno, esto es el aporte de la fluorescencia de FITC al detector de PE. ¿Cómo lo corrijo? Electrónicamente, el equipo le digo que corrija, que robe, que reste, gracias, que reste ese porcentaje de fluorescencia en FITC al detector de PE, que es sacar esta porción de detector, de fluorescencia. ¿Tá? Entonces, cuando saco esta porción de fluorescencia, ya todos los eventos son FITC positivos, PE negativos. ¿Bien? Ahí, por eso se llama compensar. Compenso ese error. ¿Bien? Lo mismo tengo que hacer con cada uno de los detectores. ¿Ok? acá para profundizar un poquito más en el concepto, ¿alguien tiene alguna pregunta? No. Para profundizar un poquito más en el concepto, ¿sí? Porque me parece fundamental que entiendan el proceso y que entiendan qué pasa si uno no lo hace correctamente, ¿no? Entonces, si nosotros aplicamos la compensación correctamente, que es esta situación, ¿no? La fluorescencia en el canal de P de las células positivas, o sea, en el eje de la CI, la fluorescencia de las células positivas para FIT-C, la media va a ser igual para las células positivas y para las células negativas. ¿Sí? Ahora bien, en una situación donde no compensé o compensé poco, esa situación se llama subcompensación, porque le puse un número que no me alcanza para compensar. Fíjense que, bueno, ya vemos que está horrible porque vemos positivos donde no existen, pero además, si nosotros analizamos la media de fluorescencia en PE de esta población, es mucho más alta que la media de fluorescencia de la población negativa. Lo mismo acá, la media de fluorescencia de la población positiva para FIT-C, la media de fluorescencia en PE, que estoy hablando, ¿sí? Es mayor que la media de fluorescencia en PE de la población negativa. Sigo estando en una situación de subcompensación. Cuando me paso, le doy un número muchísimo más alto del que necesito para tener una compensación correcta, estoy en situación de sobrecompensación. Y si analizamos la intensidad de fluorescencia en PE de la población positiva para FIT-C, vemos que es menor que la intensidad media de fluorescencia para la población negativa para FIT-C en el canal de PE. ¿Lo ven todos? Bueno, esa es una estrategia, esa es la estrategia para realizar una compensación correcta. ¿Sí? Uno a veces lo hace medio a ojo y dice, ah, sí quedaron lindas. Y se queda con eso. Pero bueno, los libros nos dicen que tenemos que mirar el valor de intensidad media de fluorescencia. Si quieren interrumpir, hacer comentarios. Claro. Siempre, siempre. Para compensar necesitas una población negativa y una población positiva. Para hacer este trabajito de mirar la intensidad media de fluorescencia en el otro detector, ahora lo haces con compensación automática. La realidad es que el aparato lo hace por vos. Pero si lo tuvieses que hacer vos solita tu alma, tenés que mirar los valores de intensidad media de fluorescencia en el detector que vos estás corrigiendo en la población negativa y en la población positiva. El aparato es lo que hace habitualmente por nosotros. Estás metiéndome en un berenjenal. Porque tenés al fundamentalista que te dice, no se toca la compensación automática, y tenés a las Florencias que te dicen, mira tu experimento. Muchas veces la compensación automática es un muy buen punto de partida, pero algún toquecito, y más si sos obsesiva, le vas a hacer, como para corregir. Y si vos seguís este criterio, ujú, ujú, exacto. Bueno, lo siguiente, sí, ¿no? Esto es lo siguiente, importantísimo, y les pongo el obligatoriamente como mandatory, ¿sí? Tus controles de compensación, porque vieron que en el diseño de experimentos nosotros tenemos los controles para el equipo, ¿bien? Nuestros controles para realizar la compensación tienen que ser lo más brillantes posible y obligatoriamente igual de brillantes que los de mi muestra. O sea, ese es mi punto de partida. Tienen que ser igual de brillantes que el de mi muestra o más. Y con eso voy a tener una compensación exitosa. ¿Está bien? Y además, como yo les dije, tenemos que tener una población negativa como para hacer este trabajito de comparación entre medias. Otro punto que tenemos que tener en cuenta, y esto es al momento de hacer el diseño de nuestro panel, es que un fluoróforo muy brillante y que derrame sobre el canal que tiene al lado de manera significativa, voy a tratar de evitar esos marcadores, los que son detectados en este que recibe mucho derrame, para ver poblaciones con bajo nivel de expresión, ¿sí? Yo a tenue le digo bajo nivel de expresión. Si yo tengo una población, un marcador de activación, un marcador que tiene muy bajo nivel de moléculas en la superficie de la célula, bueno, me tengo que enfocar en ese, tengo que darle prioridad a ese en el diseño de mi experimento y acomodarlo en el detector que menos derrame espectral reciba. ¿Está bien? De esa manera voy a minimizar el error. Voy a poder tener la mayor sensibilidad para observar la variación en estas moléculas. ¿Bien? Solo para que vean, en un equipo de 3 láseres y 8 detectores, este sería nuestro canto, ¿no? De repente, que tiene 3 láseres, la disposición de los distintos detectores, ¿sí? Y acá tenemos el espectro de emisión de FITC que se derrama en el PE. Bueno, estas cosas nosotros las tenemos que manejar para el diseño de nuestros experimentos. Porque esta es la base que después nos permite decir que nuestras observaciones son reales. Y como les dije, nosotros ya no trabajamos con 2 fluorocromos, trabajamos con 8, 10, 12. Acá les muestro un panelito de 10 fluorocromos de un experimento de una de mis becarias, por ejemplo, ¿no? Lo agarré así como al azar, para que vean que nosotros terminamos teniendo lo que se llama una matriz de compensación. Cada uno de estos numeritos es el valor de fluorescencia que le estoy sacando al detector con respecto a el otro fluorocromo. ¿Está bien? Entonces, cada detector va a tener cada experimento va a tener su matriz de compensación. Entonces, algo importantísimo también a tener en cuenta es que, bueno, nosotros hicimos nuestros controles divinos, alta intensidad, los corrimos, todo perfecto, población negativa, población positiva. Corrimos la compensación automática, nos dio bárbaro, la dejé así, no toqué nada. Pero hay un marcador que me quedó medio alto y no me gusta y le quiero bajar el voltaje. Bueno, si le bajás el voltaje, tenés que compensar de vuelta. Si una vez obtenidos los valores de compensación, no hay que alterar los voltajes del fotomultiplicador. ¿Por qué? Ustedes ya me pueden decir por qué. Estoy cambiando la escala, estoy cambiando la intensidad de fluorescencia que estoy viendo, ¿sí? Entonces, estoy cambiando la sensibilidad con que ese detector va a ver al fluorocromo. Zosia se los explicó, maravilloso, que levanta el electrón y levanta el voltaje, está buenísimo. si yo le aumento la sensibilidad a un detector, se la aumento para ese fluorocromo, pero también se la aumento para el derrame espectral que va a recibir. Con lo cual lo tengo que corregir de vuelta porque el número me cambió. ¿Se entiende? Entonces, regla de oro, no cambien los PMT. No será la figura del paper, pero bueno, salen los resultados. Y obviamente, ya sabiendo de qué se trata, toda esta situación de derrame y demás, esta matriz de compensación se va a aplicar a este conjunto de fluorocromos con estas células en particular. ¿Está bien? No puedo pasar esta matriz de compensación de linfocitos a monocitos. ¿Por qué? Porque el monocito tiene una mayor autofluorescencia, mayor pegado inespecífico, porque tiene un montón de receptores FC, por ejemplo. ¿Está? Entonces, haré mi setting experimental, haré mis controles, y si tengo células con autofluorescencia similar, o sea, voy a hacer el mismo experimento varias veces, quizá la misma matriz de compensación me sirva. Igual lo tengo que probar. ¿Está bien?
Si por ejemplo estás trabajando con una cinética, ¿no? Entonces tenés diferentes días y capaz que hay bastantes días de diferencia entre experimento y experimento. Vas al citómetro, copias el experimento, los mismos voltajes que estás usando, pero quizás hay poblaciones que te cambien, digamos, un porcentaje de población, o sea, la intensidad de esos fluorocromos va a cambiar, incluso la celularidad puede cambiar también. ¿Aconsejás de tocar la matriz de compensación para cada uno de estos días o usar la misma matriz para todo?
Bueno, eso va a depender de tu experimento. Por ejemplo, si vos estás estimulando linfocitos 5 días con un antígeno que vos esperás una respuesta importante, vas a tener cambios morfológicos, cambios en autofluorescencia, cambios en receptores FC. No va a ser la misma célula a día 0 y a día 5. Sí, quizás es la misma célula a día 0, 1 y 2, y quizá 3. Entonces, quizá la misma matriz te sirve para los primeros tiempos. Vos tenés que conocer tu experimento y tomar ese tipo de decisiones.
O sea, es válido de última cambiar la matriz de compensación, digamos. Si me cambió mucho la celularidad o la expresión de los marcadores.
Es válido, siempre y cuando vos tengas un marco teórico que te apoye, ¿no? En nuestro caso, lo que hacemos es estimular, ponele, Mycobacterium, hace que las células exploten de estimuladas. Pero las células en medio van a seguir estando quiescentes. Entonces, nosotros trabajamos siempre con la misma matriz de compensación, pero al momento de analizar, tenemos en cuenta que las células estimuladas tienen otras características y lo reflejamos en el análisis. Pero eso es porque venimos conociendo las células de añares, sabemos cómo responden y nuestro marco teórico nos lo permite. Si estás empezando de cero con una línea, células que no conoces, estímulos que nunca utilizaste, bueno, apóyate en los que saben o en los papers. Entonces, antes de largarte de hacer el experimento, tenés que pensar en todo el setting del equipo, pero también en lo que vos esperás de tu experimento. Y eso solamente lo podés conseguir vos juntando información de colegas, de gente que haya trabajado con esas células, esos estímulos, de papers. ¿Está? No, de nada. Mucho no te contesté, pero porque es muy marco dependiente. No entiendo, vos decís dos poblaciones de una misma mezcla que querés por un lado analizar una y por otro lado analizar otras. Es como si tuvieses dos células distintas. Podés hacer que con los Cytec lo podés hacer tranquilamente, podés hacer una de convolución, lo llaman eso, pero no importa, podés hacer una compensación para la población A y compensación para la población B. Me estás metiendo en otro berenjena. Porque hay gente que dice que hay que utilizar bits para todo. Y hay gente que dice que no. Bueno, otra vez, yo estoy en el intermedio. Porque uno va buscando la mejor situación en realidad. Uy, estoy haciendo lío. No, bueno, eso sí, lo tenés que hacer. No, lo tenés que hacer. Bueno, total, total. Eso es válido. Si te funciona para tu experimento, está perfecto. Hay veces que no. Hay veces que, y especialmente con los espectrales, lo que nosotros estamos viendo es que como el espectral te mete en la deconvolución la autofluorescencia, ¿no? Está bien lo que digo. Ah, ok. No, me mirás seria y digo, el espectral en la deconvolución te mete el factor autofluorescencia y la bit no tiene la autofluorescencia de tu célula. Entonces, nosotros lo que hacemos es, para los marcadores donde podemos poner células, hacemos controles con células, y en marcadores problemáticos que tienen baja intensidad, que tienen bajo nivel de expresión, baja densidad, que quizá tiene un alto nivel de expresión, pero baja densidad en la célula, ahí metemos bits. Y en ese caso, haciendo un mix, nos funciona mejor que con células o con bits. Espérate con el micrófono.
Me levanto ayer, que no. No, que para el unmixing tú utilizas el control negativo, la Nestain, para hacer el unmixing.
Claro.
Pero no necesitas que sean tus células. Cuando tú haces el unmixing, igual que la compensación, lo que necesitas es tener el espectro del fluorocromo. En la charla luego de buenas prácticas mostraré ejemplos. O sea que tú puedes tener perfectamente, que utilices una cosa u otra va a depender de... Si puedes hacerlo en células, pues te vas a ahorrar luego problemas porque te estás asegurando que como son las mismas células que tu experimento, vas a tener exactamente el mismo espectro.
Claro.
Eso no siempre es posible porque hay veces que trabajamos con muestras que son pequeñas y que la expresión del marcador que queremos necesitamos, si trabajas con animales, igual matar 50 animales, no es práctico. Entonces las bits ahí pueden ser, pero hay consideraciones que tienes que tener en cuenta. Pero no tiene por qué ser el mismo Nestain. O sea, tiene que ser el mismo Nestain de tus negativos y tus positivas, eso sí, pero no el mismo que tus células.
Bien. Bien. La aclaración, nosotros estamos haciendo espectral desde hace poco tiempo. O sea, no soy experta en espectral, así que gracias Lola por la intervención. Quizá esto lo tendría que haber explicado antes, pero básicamente es de qué se trata el derrame espectral. Y me gustó este paper, si es que lo quieren mirar, es bastante árido, pero explica muy bien de qué se trata, ¿no? Centrémonos en este gráfico, donde tenemos bits marcadas con APC67, que están perfectamente compensadas. Si ustedes ven en el detector, en este detector QD800, la media de todas las poblaciones está divina, ¿no? Pero fíjense este efecto de spreading, de desparramo, ¿no? ¿Cómo? Sí, lo que pasa es que el derrame es derramarse en el... Es como que tengo un intríngulo. Claro, este es el error, pero es una cuestión más de cálculo y de física, ¿no? A mayor intensidad del marcador, tenemos mayor despliegue dentro del detector que está recibiendo ese derrame. Y aunque yo compense correctamente, eso va a seguir ocurriendo, ¿sí? Entonces y a mayor intensidad, como vemos acá, a mayor intensidad, mayor derrame. ¿Está bien? Mayor spreading, no derrame. Entonces, esto es algo que nosotros vamos a tener que tener en cuenta al momento de diseñar nuestros paneles. ¿Sí? Sí. Acá tenemos otro ejemplo de dos fluorocromos, el Percipi 65.5 y el BUV 737, ¿no? Y vemos cómo en el detector del BUV 737 el desparramo generado por Percipi 65.5 es importante. Y fíjense que el espectro se mete un montón en el detector. ¿Tá? Yo lo corrijo eso, yo corrijo, compenso, pero aún así veo este desparramo que me hace el detector. Entonces, y fíjense, por el contrario, dos fluorocromos que no se solapan tanto, que el Percipi 65.5 no interfiere tanto, no se mete tanto en el detector del BUV 805, fíjense la resultante, ¿sí? No tengo este efecto de trompeta porque tengo los detectores más alejados. Entonces, cuando yo tenga que elegir un marcador para un anticuerpo, para un marcador, para una proteína que tiene baja expresión, por ejemplo, no voy a elegir nunca esta combinación de anticuerpos. Porque ponele que yo la ponga a mi... A ver, no, perdón, pensé que había dejado una diapo. Imagínense si yo utilizo para marcar, no sé, CD25, que es un marcador que está en tiene muy poca expresión la célula quiescente. Si marco CD25 con BUV737 y lo combino con un CD4 Percipi, 65.5. El CD25 me va a caer por acá, ¿sí? Y quizás me moleste este spreading que me está haciendo el fluorocromo Percipi 65.5. Me va a bajar la sensibilidad. Bien, la sensibilidad y la resolución en cuanto a marcadores de baja intensidad. Entonces esta combinación me sirve, porque si lo combino con un CD8, veo claramente la población CD8, veo claramente la población 66B, perfecto. ¿Está? Tengo el efecto, convivo con él, lo utilizo. Pero tengo que tener ese cuidado de no utilizar estos canales que reciben mucho derrame para marcadores de baja intensidad. Entonces, para mi experimento, que lo corro en un equipo específico, voy a tener una matriz de derrame, que no es ni más ni menos que el número que aporta cada fluorocromo a los detectores donde derraman, ¿sí? Con lo cual, intuitivamente, yo podría decir que la matriz de derrame sería la inversa de la matriz de compensación, ¿no? Si yo al compensar corrijo lo que se está derramando, este numerito de derrame sería lo que tengo que compensar después. ¿Está bien? Así que bueno, nosotros habitualmente esto no lo hacemos, de calcular el derrame. No sé ustedes, chicas, si en sus laboratorios lo hacen. Porque directamente vamos a generar nuestra matriz de compensación para cada experimento. Pero la generación de estas matrices nos ayuda a conocer nuestro panel y a tomar decisiones, como les digo. Por ejemplo, cuando tengo fluorocromos con alto solapamiento, marcar poblaciones que sean mutuamente excluyentes. Por ejemplo, CD3 con CD19. Entonces, no se molestan, todo fantástico. Otro tema que me gustaría charlar con ustedes es el control de FMO, que tiene que ver con todo esto, ¿no? Con el derrame espectral y con la utilización, la utilización de varios fluorocromos y el análisis de cómo diferentes fluorocromos aportan a un detector. Entonces, Fluorescence -1 se trata de eso. Cuando estoy trabajando con un panelito, centrémonos en el panelito de acá arriba, voy a hacer controles, ¿no? Los controles de compensación, unstain, todo pero también voy a hacer los controles FMO, donde voy a incorporar todos los marcadores menos 1. Ese menos 1 es el marcador que a mí me interesa. Y por lo general es un marcador problemático. Un marcador de activación, un receptor que se induce luego o que se inhibe luego de la activación. Cosas donde yo quiero poner un cutoff lo más prolijo posible. ¿Está bien? Entonces, fíjense que tengo mi muestra marcada con todos los marcadores y tengo el control FMO sin el marcador que a mí me interesa estudiar. ¿Está bien? Si corro la muestra sin marca, voy a poner un cutoff. Si corro la muestra, el control FMO, fíjense cómo se comporta mi cutoff. Cambia. Bien, mi sitio desde donde arranca la positividad se levantó. Entonces, si yo utilizara solamente muestras no marcadas, tendría un montón de falsos positivos. ¿Lo ven? Con lo cual, hacer este control me permite tener un cutoff mucho más fidedigno y ahora sí poder ver claramente mis células positivas. O sea, yo no lo hubiera usado para un CD4, yo lo hubiera usado para un CD69, CD25, un PD1. ¿Bien? Sí.
Entonces, como tú mencionas, no lo hubieras usado para un CD4, entonces los FMO no hay que usarlos siempre o para todos los colores,
Exacto.
sino que solamente en base a si el marcador es difícil o no debe.
Exacto.
Y si, por ejemplo, hago el mismo experimento siempre, si yo ya calculo mis cutoff, ¿puedo correr el siguiente experimento sin necesidad de hacer los FMO?
En Sudamérica sí.
¿Cierto? Porque salen caros los anticuerpos.
Todo depende de la repetitividad, de cuán de cuánto de cuán estable sea tu experimento, ¿no? Obviamente. Pero sí, porque si no estás marcando 15 tubos para ver uno.
Yo tengo una pregunta con respecto a cuando uno delimita el gate según el FMO. Porque según la imagen esa, digamos, está marcado el gate del FMO, pero hay unas células que son positivas, que están más allá, digamos, del gate.
Bueno,
Entonces, uno,
sí.
digamos, cuando lo quiere hacer, a mí me pasa también cuando lo hago. Entonces, digo, uno lo tiene que hacer como a ojo, digamos, o considerar hasta la última célula, hasta dónde, digamos, uno debería marcar el gate según el FMO.
Bueno, volvemos a base. Todo depende de tu experimento. Todo depende de la frecuencia de las células positivas que vos esperás.
Sí.
Depende de la intensidad de las células positivas que vos esperás y de cuán benévola seas vos en tu experimento. Entonces, pasando por esas 3 fases de decisión, vos la tenés que mantener. No es que este experimento, pero este se lo corro porque está nublado, este se lo corro porque hace calor. No.
Mantener el criterio.
Mantenés el criterio y a partir de ahí, como a ciegas, seguir el análisis. Vos, si estás esperando frecuencias de 40% y esas poquitas positivas son un 0,1, no te va a mover el amperímetro. Si en esas poquitas positivas estás dejando 0,1 pero tu positivo es 0,3, 0,2, y bueno, tenés que revisar tu estrategia de gating y de número de células con las que estás trabajando. Entonces es muy experimento dependiente. Yo sé que hay trabajos donde hacen cálculos estadísticos dependiendo de la frecuencia esperada, cuántas células tenés que marcar y adquirir. Está muy protocolizado para la terminación de CD34, por ejemplo, en sangre periférica, tanto la marcación como el análisis y el número de eventos que tenés que adquirir, porque son células que están en muy baja proporción, muy baja, y vos tenés que verlas claramente y cuantificarlas de la manera más eficiente posible. Entonces, todo es muy contexto dependiente, pero a grandes rasgos, si vos estás con un experimento nuevo, ponés los criterios después de un trabajo importante, no es que decís, lo pongo acá y de acá arranco, no. Vos tenés que ver cómo expresa tu marcador, cómo se comporta tu anticuerpo, cómo lo acomodaste en tu panel. Son varias cosas que tenés que tener en consideración para tomar esa decisión, de decir, bueno, a partir de acá, esto es mi positivo. Y de ahí no moverte. No, de nada. Eso con respecto al FMO. Algo también que me gustaría introducirlos es en la utilización del dump channel, ¿sí? Utilizar un canal para tirar a la basura o meter ahí todas las poblaciones que no quiero analizar. Y de esa manera, en el análisis siempre, ¿no? Tener más pura la población de interés. ¿Ok? A eso llamamos dump channel. Acá les puse como definición que es el proceso de exclusión intencional de células en un análisis intencional a que yo lo incorporo en mi protocolo, ¿está bien? Lo incorporo en mi experimento, lo trabajo y de esa manera me saco de encima las poblaciones que me están molestando en mi análisis. Se usa un marcador o una combinación de marcadores, por lo general son marcadores de linaje, ¿sí? Nos muestran linaje de T, B, NK, monocitos. Además de la tinción de viabilidad. Eso seguramente lo van a ver en el transcurso de los trabajos prácticos de los wet lab, la importancia del marcador de viabilidad en nuestros experimentos, ¿no? Se puede utilizar uno o varios canales para marcar las poblaciones no deseadas, incluso las células muertas. Puedo utilizar todos los marcadores para un mismo canal, o sea, marcar todas las poblaciones que no me interesan con un mismo fluorocromo. En ese caso, el proceso de compensación es más sencillo. ¿Está bien? Porque tengo todo ahí con alta intensidad en ese canal. Pero también puedo hacer una exclusión secuencial. Y otra vez lo mismo, volvemos a qué necesito yo para mi experimento. Entonces, utilizamos diferentes canales para cada uno de los linajes y vamos haciendo un análisis secuencial de las poblaciones. Acá yo les traje un ejemplo, que este paper me gustó mucho, para ejemplificar, ¿no? Acá están buscando células CD34 positivas, que son estas y estas. Estos paneles de arriba son utilizando dump channel con un solo dump channel para todas las poblaciones. Entonces, acá marcan 3, 14, 20 y 56, ¿sí? Todas las poblaciones que no quiero, las marco con el mismo fluorocromo. Y tomo las que son negativas para todos esos fluorocromos. Entonces, a partir de ahí hago mi análisis y me resulta en 2.18 células CD34 positivas, que esta que yo les conté, que son las STEM en sangre periférica. Y en este segundo protocolo, lo que hacen es una eliminación secuencial de cada una de las poblaciones. Entonces, marcan CD14 con un fluorocromo, 20 y 56 con otro, y CD3 con otro. Entonces van de a pasos eliminando las diferentes poblaciones. Hacen su análisis y como resultante tienen 3.33% de células CD34 positivas. Entonces concluyen que para este setting experimental, la utilización de down channel en diferentes canales y haciendo un análisis secuencial de eliminación de las poblaciones que no me aportan, mejora la resolución de mi experimento. ¿Está bien? Así que bueno, me pareció interesante una estrategia más en la utilización de todo lo que es estrategias para mejorar nuestros experimentos. Bueno, y por último, re bien. Por último, simplemente para que vean una comparativa de la disposición de detectores. Abajo tienen un citómetro convencional, los detectores para el láser en violeta, son las barritas, ¿ven? Y cómo los diferentes fluorocromos aportan a cada uno de los detectores, mientras que en un citómetro espectral, para el láser violeta hay 16 detectores, el mismo espectro de fluorocromos, vean cómo es captado en diferentes rangos de longitudes de onda. Entonces, finalmente, que ya lo van a ver en las charlas de espectral, ¿no? Pero como para introducirlos, cada uno de los fluorocromos los vamos a ver como una huella. Cada fluorocromo va a generar una huella. Esa huella está dada por la fluorescencia vista, detectada por cada uno de los detectores cuando ese fluorocromo es excitado por los diferentes láseres. ¿Ven? El FIT-C detectado por los detectores del láser violeta, azul y rojo. Entonces se genera una huella para cada uno de los fluorocromos. Y para que vean que es totalmente diferente, yo no les voy a hablar, hola, justo llegó María, no les voy a hablar de espectral, pero para nada, pero simplemente para contrastarlo con la citometría convencional y vean que es otro pensamiento, otra lógica, ¿bien? ¿De qué se trata el proceso de deconvolución? Porque uno habla siempre de compensación, la tiene súper incorporada, bueno, el que aporta al otro detector se lo resto, ya está, lo resté, lo que ve ese detector es real. En la deconvolución te explota la cabeza, porque vos tenés, por ejemplo, un espectro que es la suma de, en este caso, 3 fluorocromos. Ese proceso de deconvolución lo que tiene que hacer es calcular cuál es el aporte de cada uno de esos fluorocromos a este espectro. Y de esa manera, conocer cuál es el nivel de expresión de cada uno de los fluorocromos y por ende del anticuerpo que está pegado y por ende de la molécula que se está expresando en esta célula. ¿Está? Y eso lo van a ver maravillosamente con María. Pero esto es simplemente para contarles que sistema de fluidos, sistema óptico, son iguales en los dos equipos, y el sistema de detección, van a ver que tiene ciertas variaciones, pero es básicamente detección de luz en rangos de longitudes de onda, pero después todo lo que es el análisis de la participación de cada uno de los fluorocromos en una señal es totalmente diferente. Y para poder utilizar equipos espectrales, tenemos que conocer cómo es ese proceso, porque cuando tengamos un problema vamos a tener que resolverlo, y la única manera de resolver los problemas es conociendo la base teórica con la que estoy trabajando. ¿Bien? Así que bueno, con eso termino. Muchísimas gracias. Y bueno, preguntas, comentarios.
Creo que ya lo hablamos, pero bastante concisa, ¿sí? Dice, ¿recomienda hacer la compensación automática? Si llegamos a hacer compensación automática una vez hecha, pero antes de pasar las muestras, ¿se pueden modificar los voltajes?
Se puede modificar la compensación automática en base a lo que hablamos hace un rato, ¿no? En base a tu experimento y demás, que seguramente vas a tener que hacer una pequeña corrección, pero es más bien de obsesivo compulsivo que necesario para el experimento. Ahora, mover voltajes, ya dijimos que no. ¿Por qué? Porque si vos movés el voltaje de un detector, lo que implica eso es cambiarle la sensibilidad de detección a ese detector. Si le cambio la sensibilidad de detección, se la voy a cambiar para el fluorocromo que yo quiero mejorar, pero también voy a cambiar la sensibilidad de detección de los fluorocromos que están aportando en su derrame a ese detector. Bien, con lo cual, terminantemente prohibido, después de hacer todo el trabajito de compensación, cambiar voltajes. Bueno, muchas gracias.